Hello everyone. In this video, we'll see about acid-fast staining, also called as Ziel-Nielsen staining or Zn staining. So this was discovered by Ehrlich, who found that after staining with aniline dye, tubercle bacilli resist the decolorization with acids. Okay. So this acid-fast staining we do for sputum sample to detect the Mycobacterium tuberculosis. Okay, so this was modified by Zeal and Nielsen in the uh, coming days. Okay, so which was you nowadays it is in practice. Okay, so it was found that the tubercle bacilli which resist the decolorization with the acid. So that's why it is called as acid fast staining. So we'll see about the principle of staining. So what is the principle here? The acid fastness has been ascribed to high content and variety of the lipids, fatty acid and higher alcohol found in the tubercle bacilli. So this tubercle bacilli has high content of lipid, okay, and fats. So when you add the alcohol, acid alcohol mixture or this 20% sulfuric acid, so which resists the decolorization. So this the lipid which is present in the Mycobacterium tuberculosis is peculiar, so which has the high molecular weight hydroxy acid wax containing the carboxylic group, that is mycolic acid, so which is present in the free state. Okay, so acid fastness mainly depending upon not only on the lipids, so it is also depending upon the integrity of the cell wall. Okay, so as you know, the Mycobacterium cell wall is complex when compared to normal gram positive or gram negative bacteria. Okay, so principle is based on the presence of high content of the lipid, fatty acid and the alcohol, so which resists the decolorization with the acid. Okay, so we'll see the procedure and what all reagents we required. Here to do the Zn staining or acid fast staining, we require strong carbylfuscin stain, acid alcohol or 20% sulfuric acid, and we want malachite green for the counter stain. Okay, what we will do, we will prepare the smear first, let it be air dried. Okay, so for that fixed smear, okay, we'll heat fix and we add the strong carbylfuscin. Okay, so what we have to do, so we are keeping slide on the rack and we'll add the stain, right? So we'll take one cotton ball and we'll add some alcohol to that, and we'll fire it and below the slide. Okay, so if you have a spirit lamp, it is good. So if you don't have, you can take the cotton with alcohol and you make like fire you will take. Okay, so gently you will heat from the below of the slide. So the it should just bubble should come, not the stain should not evaporate or it should not boil. Okay, just the fumes, when the fumes arises, you stop the intermittent heating. Okay, so intermittent heating means in between. So after adding carbylfuscin, we are keeping for 10 minutes, right? So in between like 5 times or 4 to 5 times we have to heat in between, so that we call it as intermittent heating. So it should be continued till 10 minutes. If the stain is dried or the stain is getting evaporated, so you add more stain. Okay, so the process of heating helps to enter the dye, that is carbylfuscin, into the bacterial cell wall. Okay, so this carbylfuscin, when you heat it, right, so from the below, so it will penetrate inside the cell wall of the bacteria. Okay, then this smear is washed with water and you add the decolorizer, that is 20% sulfuric acid, so until the color, the pink color should go out of the smear. Okay, so after that process, it may take 2-3 minutes to wash out that stain. Okay, so once that stain is washed off, when you think that the smear is colorless, so then you add the 2% methylene blue and keep it for minimum of 2 minutes. Okay, 2 to 3 minutes we say. So you will keep it and then after 2 minutes you will wash it with water, dry it and observe under the microscope. So what you will see here, so you will see the acid fast bacilli. Okay, pink color, it will be, the bacilli will be pink color with the beaded appearance. And when you add the methylene blue, the pus cell which is present in the smear will take up that color and it will appear in the blue color. Okay, so this is the procedure. What you will do, you will take the smear and cover it with the carbon fuscin. Intermediate heating technique you will do from the below, and after that you will decolorize with the 20% sulfuric acid or acid alcohol mixture. So then you will counter stain with the methylene blue and you will dry it and observe under the microscope. Okay, so this was the procedure of acid fast staining. Hope you understood the concept of acid fast staining. If you have any doubt, you can comment below. Thank you.